Thank you so much for the opportunity to speak today. Yes, I will be trying to cover my entire 4-year PhD in a 10-minute presentation, so bear with me. It will be in the spirit of the session fast. So I'm just going to jump straight into it. Right. So the liver is composed of microscopic hexagonal units called lobules, and these comprise a central vein which is surrounded by branches of the hepatic artery and portal vein, as well as the bile ductules. The hepatocytes are the main epithelial cell type of the liver and make up the majority of these lobules. And they also conduct most of the functional functions of the liver, which include, obviously, the synthesis of blood proteins, storage of glycogen, breaking down drugs and toxins. And alongside these hepatocytes, you've got the bile ductules, which are made up of the biliary epithelial cells, which you've heard a lot about in the last 3 days. So these are responsible for the collection and modification of the bile, which is produced by the hepatocytes, and helped to drain it out of the liver. So although these two types of epithelial cells are very different in their roles and morphologies, they actually originate from a shared bipotential progenitor cell with signaling activation in pathways such as Wnt and TNF-alpha driving a hepatocyte fate, whereas a signaling upregulation in pathways such as Notch and TGF-beta forcing a biliary lineage. Interestingly, though, although the hepatocytes look very morphologically similar across the lobule, they actually are very functionally dependent on where they sit along this portal to central vein axis. So this axis can be broken down into 3 zones, namely the periportal zone 1, the midlobular zone 2, and the pericentral zone 3. And because of the periportal hepatocytes being exposed to blood, which is rich in oxygen, nutrients, and circulating hormones, they specialize in functions such as gluconeogenesis and beta oxidation, whereas the pericentral hepatocytes around the central vein tend to function in things like glycolysis and lipogenesis, which is obviously the waste management side of things. So these oxygen and nutrient gradients, as well as signaling gradients, which predominantly are listed here, likely influence how these hepatocytes respond to injury and oncogenic stresses. So as you all know, the liver is one of the few organs that can regenerate itself after injury, and it does so through several mechanisms, which include the cell proliferation, recruitment and differentiation of progenitor cell types, and in certain injury cases, the epithelial cells have been shown to be able to change their cell lineage, which is termed transdifferentiation. So in this context, it's become evident that hepatocytes are actually not as fixed in their fates as previously believed. They can exhibit what is known as plasticity, and it's not known whether, before this project, whether this plasticity is characteristic of all hepatocytes or whether it's very zone restricted. And in some cases, this plasticity of hepatocytes to biliary cells specifically can help to repair damaged ducts, but it can also contribute towards early stages of iCCA. Although the hepatocyte to biliary conversion, as I say, has been recognized previously, before I conducted this project, it was unknown whether it was a universal property. So the null hypothesis of my PhD was that zonation is actually a major determinant of hepatocyte plasticity. And more specifically, I proposed that the periportal and pericentral hepatocytes differed intrinsically in their ability to undergo this transdifferentiation because they were at the different ends of the poles of these lobules. So to test this, I used two complementary approaches, the first being the development of in vivo mouse models, and the second being an ex vivo primary hepatocyte spheroid system. So, as I said, the first thing I did was generate two mouse lines, which made use of a Cree-driven system with dual fluorescent MTMG reporters, which basically means that all cells in the livers, or all cells in the mice, initially expressed a red fluorescent protein on their membranes. And when we gave them a selective estrogen receptor modulator called tamoxifen, the membranes of the cells would then turn green. So this allowed us to then label the cells of interest. And in this study, we used glutaminase 2 as the selective Cree-driving gene for the periportal hepatocytes, and glutamine synthetase, which is a Wnt-high pericentral zone 3 marker. So using immunostaining and flow cytometry and bulk RNA sequencing, I showed that the two periportal and pericentral hepatocyte lineages were distinct in both their transcriptomes and their functionalities, with periportal GLS2 lineage hepatocytes being predictably enriched for gluconeogenesis, amino acid metabolism, and urea cycle pathways, whereas the GS lineage cells showed upregulation and expression of canonical Wnt target genes and signatures of xenobiotic and lipid metabolism. So then we went on to drive a hepatocyte to biliary reprogramming in the liver. And to do this, I used what is known as the hydrodynamic tail vein injection model, which allows us to deliver plasmids directly into the hepatocytes. And the two plasmids that we used, which were from a previous publication, were the Notch intracellular domain 1, which drives the cells towards a biliary-like fate by upregulating Notch signaling, and a myrosylated AKT, which promotes the cell's survival and proliferation. So the simultaneous upregulation of these two signaling pathways induced the formation of these biliary-like neoplasms throughout the liver, and they were characterized by the formation of lumen-like structures, as well as upregulation and proliferation, which is shown by the Chi67 staining. And they also started to express markers of biliary lineage, such as SOX9 and keratin 19. So this model provided us with a robust system to ask whether the periportal and pericentral hepatocytes intrinsically are different when we give them the same oncogenic stimulus. So after treating both the GLS2 and GS MTMG mice with tamoxifen and hydrodynamically upregulating the Notch and AKT signaling, the cells that were green labeled, so the GLS2 expressing hepatocytes, demonstrated a noticeable contribution towards the formation of these biliary-like neoplasms. The cells lost their typical polygonal morphologies. They became much smaller and cuboidal, and they arranged into the duct-like structures with the lumens forming. And they also started to express SOX9 and keratin 19, as we had shown in the preliminary data. So this kind of showed to us that the GLS2 expressing hepatocytes were highly plastic, and they were able to contribute towards these biliary-like neoplasms potentially being a cell of origin. In contrast, though, the GS cells contributed very little to, if at all, to the lesions, despite clear evidence that they had uptaken the plasmids, and they were also in very close proximity to the forming tumors or neoplasms. But they largely kept their hepatocyte morphologies under the same oncogenic stimulus that the GLS2 cells had experienced. And we did quantitative analysis, which indicated that the near absence of the GS positive neoplasms wasn't due to poor sampling or poor labeling, but it actually reflected an intrinsic difference between the pericentral and the periportal hepatocytes in response to this Notch AKT-driven transdifferentiation. The green fluorescent cells were then isolated from both mouse models and sent for bulk RNA sequencing, with or without the Notch and AKT treatments. And the zone 1 cells, the Notch and AKT treatments induced extensive transcriptional rewiring with biliary progenitor cell associated genes, including the progenitor genes being upregulated, the hepatocyte ADT genes being downregulated, and there was also pathways including cell cycle, DNA repair, chromatin remodeling, and mitochondrial functioning being enriched. And in the GS lineage, there was much more restrained responses, and it was more stress responses that we were seeing in these cells rather than a reprogramming. So while these in vivo experiments showed a zonal hierarchy, we then went on to looking at in vitro hepatocyte culture to see if the zonal features were preserved outside the body and whether we could control them. As being shown in literature, 2D culture of hepatocytes is impossible. It's not impossible, but it's very difficult. They tend to lose the plot after 96 hours. They become very mesenchymal and are not hepatocytes at all anymore. So we then moved on to looking at three-dimensional cultures, and these showed that they were able to maintain the functionality and morphologies of the hepatocytes longer term, with albumin secretion being maintained, glycogen production and storage, as well as the cytochrome activity. So this, the spheroids were also shown to be able to maintain zonation to some degree, and we were able to modulate it by adding certain supplements into the media to drive it towards either a periportal or pericentral-like phenotype. So that brings me to the conclusions of the study, which were basically that the hepatocyte plasticity is not equal across the lobules, with the periportal hepatocytes being more plastic, and the pericentral hepatocytes being retaining a hepatocyte identity. The second is that the three-dimensional hepatocyte spheroids can recapitulate aspects of the hepatocyte functionality and zonation, and they can be shifted towards a periportal, pericentral-like state, depending on what the media is supplemented with. I'm currently trying to build on this by trying to look at whether the Wnt signaling in the pericentral cells is the protective factor, which we kind of got from some of the signaling, the sequencing results. So to do this, I'm just either trying to dampen the Wnt activity in the central vein cells using different inhibitors. And I'm also going to potentially look at globally enhancing the canonical Wnt signaling to see if it increases the resistance of the other cells. So overall, the hepatocyte zonation is not equal across the lobule, and it's a key factor of its fate during injury. So better understanding this and modulating this could transform the prevention and management of intrahepatic cholangiocarcinoma and other zonal liver diseases. So thank you very much. Unfortunately, I've got a plane to catch. So if anyone has any questions, you can either ask me on the AMMF app, or you can visit our very nice guest lab website, theguestlab.com, and we will be happy to answer any questions. But thank you very much, everybody.